Because we're going to talk about chapter 20 and a little bit of 1019, especially some basic overview stuff of viruses, bacteria, and prions. So viruses are extremely small. So this is a red blood cell. Here's bacteria. Blow that up, and you can see these tiny little viruses down to the little poliovirus and bacteriophages. So viruses can contain DNA or RNA, and they can have single-stranded DNA or double-stranded. Really all they are is the nucleic acid, DNA or RNA, surrounded by a protein coat, which is the capsid. Some can have an envelope outside the capsid, which is usually made of the host membrane. So bacteriophages are a special kind of virus that only infect bacteria. So here, this bacteriophage, a little alien dude, is sitting on top of the bacteria, and he's injected his DNA. He then convinces that bacteria to create more viruses. The bacteria create the protein coat and the DNA, and then the virus come out and destroy them. So viruses can't reproduce on their own. HIV, which makes the AIDS virus, is called a retrovirus, and what that means is it contains RNA. They infect the white blood cells, and it has what's called reverse transcriptase, which is actually an enzyme that can use RNA to make DNA, which is backwards than what it usually is. There's two different cycles, lytic versus lysogenic. I think of it, ly dies. So with lytic, you have the bacteria, or sorry, the virus injects the DNA, and then inject it really over here, in there, and again, the bacteria are convinced to make proteins and DNA or RNA for the virus. They hold bunch of viruses, and they lyse out or burst out of the cell. Versus lysogenic, we have the DNA. It actually becomes part of that bacterial DNA, which is called a prophage. And so you have this prophage, which is just viral DNA and bacterial DNA put together. It replicates, keeps replicating, and we have daughter cells with that. Prions, those are actually just proteins, so it doesn't even have the DNA. They're misshaped proteins, and they can change the shape of other proteins that they contact, so you can get aggregates, and this is what we believe causes mad cow disease. Along with, so we have these aggregates, other scrapie, chronic wasting disease, which you find in deer, and humans are the Creutzfeldt-Jakob disease. So, a little bit about bacteria. One-celled prokaryotes, they reproduce by mitosis, which is called binary fission, one cell becoming two. They can reproduce every 20 minutes. So overnight, we get over 100 million. That's why you can start seeing colonies. They're dominant. They pretty much cover everywhere on Earth, and they're incredibly diverse. It's said that if we actually classified them right, that each type of bacteria would have its own kingdom. There's a lot of different ones. And so the bacterial genome has a single circular chromosome. They're always haploid, so one set of the chromosome, and it's naked DNA. There's no histone included. There's about 4 million base pairs, 4,300 genes, which is 1/1000th of the DNA of a eukaryote. So how can they be so diverse? Interesting. So the way that they replicate is you have an origin site, and then you get your replication fork, and you can see the semi-conservative model, light blue and dark blue, as it keeps going, and at the other end there's a termination, and you have two different ones. So offspring are identical. So if offspring are identical, again, where's the variation? So the way that they have variation is it could be spontaneous, just like with us. There could be a mutation of DNA. They also could have transformation, which is DNA fragments from the environment that bacteria can take in. They can go through transduction, which is when a virus introduces DNA. They can go through conjugation, which is a form of bacterial sex, or they can have transposions, which we'll talk about in a little bit as jumping genes. So, spontaneous mutations, they are rapidly reproducing, and so you have exemplary E. coli, which are in our colon, and there's about 2 billion new E. coli each day. And so if there's only 1% or one mutation per 10 million replications, that would be 2,000 different bacteria that are developing mutations. 4,300 genes, you could have 9 million mutations per one human host. So there's a lot that could happen. Transformation. So we actually did this with the bacteria. So yes, they will pick up foreign DNA wherever it might be hanging out, and so they have surface proteins that are specialized to take up this DNA, and they'll import bits of chromosomes from their other bacteria, so they just take it, and then they're able to express new genes, a form of recombination. And so this is get the virus. So plasmids are the small pools of DNA, which have 5,000 to 20,000 base pairs. They are self-replicating. They can carry genes. So these are all little plasmids. That's the circular chromosome. It can have 2 to 30 genes, and they also usually will carry genes for antibiotic resistance. And these can be exchanged between bacteria. Ooh, bacterial sex. So that they have a very rapid evolution, and they can be imported also from the environment. So the genes for antibiotic resistance we are plasmids, and they have a role in rapid evolution, and it can also spread antibiotic resistance, where bacteria are no longer affected by our antibiotics that we take. So we first started seeing this in the 1950s, and when dysentery, which causes like a lot of diarrhea and stuff, wasn't responding to antibiotics. Now we're seeing that this is a worldwide problem, and this is why you need to take your antibiotics fully so that you don't have these bacteria going around still. Another way that they can reduce is through transduction, and so viruses have DNA, and you see produce more, but we have some of these that we are in as lethal, and that actually can become part of the genome. Conjugation is bacterial sex, so they actually take their pili. See another picture here, and they actually can transfer a plasmid from one to the other. So it's direct transfer of DNA. Not letting you see this very well, but these are transposons, which are jumping genes, and they can actually move from one place to another. So they can actually move the new genes to a different site, which is pretty remarkable. So I'm going different places. Alright. So the next thing we're going to talk about is biotechnology. How are we using all these things in technology today? So we have all kinds of different parts. Basically, it's all summed up in genetic engineering, which is a manipulation of DNA. And so you have to have different tools to be able to work with. So we're going to be looking at our different tools. So we can look up crime scene analysis. We can make insulin. We can modify rice. We can alter DNA in humans. We can do phylogenetic trees and try and figure out. We can change the color of this poor piggy and rat. So why would we do all these? What are we doing? So reasons why we're doing a lot of this is treating diseases, and so detecting viruses, gene therapy, being able to purify genes, pharmaceuticals, so we can actually produce insulin, human growth hormone, crime scene analysis, and genetically modified food. We can introduce vitamin A into rice, so it's better for you. We can make corn and different things resistant to bugs, and we can get bacteria to clean up toxic waste for us. And so we basically do this. Let's see, I'll talk about these a little bit, but you can see several different parts that we get bacteria to grow things for us. So first we'll start off with genetically modified foods, which are GMO, and these will enable plants to produce new proteins. So we can protect crops from insects, which the corn produces a bacterial toxin that kills the corn borer, which is the caterpillar. We can extend the growing season. So strawberries with the anti-freezing gene that actually came from a fish. We can improve the quality of food. So we have rice that produces vitamin A. So how do we get plasmids to help us? Remember, those are the little circular pieces of small parts of bacteria DNA. So we insert that new gene into the plasmid. We insert the plasmid into the bacteria, which is a vector, and the bacteria now expresses the new gene. Bacteria make the new protein, so we cut the plasmid, and those are with restriction enzymes, not actual scissors. We have the gene from the other organism, and we now have recombinant DNA, glue it all together. It's now inside the bacteria, and every time this bacteria reproduces, it makes whatever new protein we want. So again, that's what we did with our transformation lab with bacteria, and that's why they were pink. And so plasmids are used to insert new genes into bacteria. We get the genes we want: insulin, lactase, so that you're not lactose intolerant, human growth hormone. We'll cut the DNA, we'll cut the plasmid DNA, insert the gene we want, glue them all together with ligase, and we get a recombinant DNA. How do we cut the DNA? So we have different restriction enzymes. We also have restriction new nuclease we found in the 1960s, and these evolved in bacteria in order to cut up foreign DNA. And so they can restrict the action of the attacking organism, protect against viruses and other bacteria. And so bacteria protect their own DNA by methylation, adding a methyl base and not using the base sequences recognized by the enzymes in their own DNA. So what do you notice about these? You're not noticing it yet. Every single one of these is a palindrome. If you don't remember a palindrome, it reads the same forward as it does backwards. Yes, this is important. I have a place where I'm going with this, but you'll have to wait till the next video.